And now we come to one of the hottest areas of research right now, the human microbiome. Scientists for many years knew that microorganisms live in an ecosystem on humans, but it was only in the last few decades we realized the profound effects our little co-pilots have on our health and well-being. What kind of microbes colonize humans? You have the whole gamut of little critters living on you. There are bacteria with four phyla dominating: Bacteroidetes, Firmicutes, Proteobacteria, and Actinobacteria. You also have archaea, with one of the main known players being the methanogen Methanobrevibacter smithii. There are also viruses, both human and many bacteriophage, and yep, there are also fungi, protists, and little animals. For the most part, all of these are helpful to your health and well-being. A fertilized egg begins sterile. During pregnancy, it appears colonization begins from the placenta, and it resembles the oral microbiome of the mother. During birth, the infant is inoculated with microbes present in the vaginal canal. Interestingly, the initial microbiome of infants delivered by cesarean section is different than vaginal birth, but this dissipates with time. Breast milk also contains milk glycans, including oligosaccharides, and these are polymers of sugars, that are indigestible by humans. These polymers promote the growth of the microbiome, selectively enriching gut bifidobacteria. So think about that. We make, or mothers make in their milk, oligosaccharides that humans can't digest. It is specifically designed to support a specific microbiome. That's pretty amazing. After infancy, the gut microbiome changes, and there is an ecological succession until a stable community appears. Two landmarks during this process are the introduction of solid food and then puberty. All right, the roles of the microbiota, an overview. The contributions of the microbiota fall into four broad categories. Nutritional function, and this is microbes helping you to digest food and provide vitamins. Barrier protective functions, and this is where the microbes restrict other microbes, potentially pathogens, from colonizing. They also help reinforce barrier functions. And then immune functions, microbes educate the immune system. They are the little sparring partners for our immune system, and they keep it on its toes. And then community function. Microbes alter the local ecosystem, and because they do this, that will help other organisms grow or prevent other ones from growing. So, where are you colonized? Here's the places. You are colonized on your dry epithelia, the skin, and this will have a very different microbiota than the moist epithelia, your mucosal membranes. The respiratory tract, the gastrointestinal tract, and the urogenital tract. Let's look at those environments. The microbiota of the skin has, a different microbiota than the moist surfaces of humans. Skin surfaces are covered in microbes, and you can see a little graphic here showing you the bacteria, which are red, the little mites that eat your skin cells, and then all the viruses that are present, and there's also some fungi. You have about 110 species are normally occurring on the typical human. Altered microbiota is correlated with disease stage, such as psoriasis, atopic dermatitis, and rosacea. The skin microbiota varies between sites. Here is some data from the Human Microbiome Project, and if you look at the proportion, these little pie charts show you different types of bacteria. So you'll see here the Actinobacteria, the Bacteroides, Proteobacteria, and then Firmicutes. And it shows you how many are there. You'll notice there's profound differences between the facial skin, then skin under the armpits, etc. And it all really depends on two things: the amount of sebaceous glands that will make the skin wetter. They'll have more fluid in those areas. That tends to help Propionibacterium species dominate, versus drier areas. Drier areas will have a greater diversity, and proteobacteria will dominate in these places. In moister areas, you'll have more Staphylococcus species. But again, note that a few types dominate. The main take-home message is the skin microbiota varies between sites. The skin microbiota varies also between people. Here we're looking at 4 different areas. The antecubital fossa, which is the little crease in your elbow, the back, the nose, and the heel. And you'll see that the proportions of all these different phylotypes is different between different groups of people. Right, so it can vary in the site, it can also vary between individuals. One of the main organisms that you'll find in almost 100% of people's skin is Staphylococcus epidermidis, and this colonizes many sites on the body. This is a helpful organism to us. It produces bacteriocins that are active against Staphylococcus aureus, a known pathogen. It inhibits S. aureus biofilm formation. It induces production of host cell antimicrobial peptides. And it inhibits excessive inflammation following an injury. Now, rarely Staphylococcus epidermidis is pathogenic. Now on to the gastrointestinal tract. Just for reference, this slide shows the anatomy of the GI tract and the function of each tissue. Note, I will not be testing you on this. I just wanted to remind you of what we're actually talking about. The first spot that we'll look at is the oral microbiota. You have about 400 different species in your mouth, and this is probably one of the higher concentration of microorganisms in your body. The teeth are a non-shedding surface, and that allows microorganisms to attach to them and to set up shop, basically. The oral microbiota plays a role in periodontal disease, such as gingivitis. It may be linked to diseases at other sites. This is somewhat controversial. Preterm birth, cardiovascular disease, pancreatic cancer. It is the most conserved microbiome between individuals. If you compare individuals, they'll have a very similar microbiome. And the plaque that sets up on your teeth will have lots of Streptococcus species on the bottom, and then there will be this secondary biofilm and then tertiary biofilm that binds to the Streptococcus that are bound to the teeth. And this can build up to high levels, such as there's this extensive microbe interactions and this coaggregation. And this is why it's so important to brush and floss your teeth, is to get rid of that biofilm. If we look at the rest of the gastrointestinal tract, going from the esophagus all the way down through the large intestine, it begins very lightly colonized in the esophagus because there's such rapid transit of material through here, to the stomach, which has a high acid concentration, which also decreases the amount of organisms, and the colonization increases as you go down. Transit is fastest at the beginning of the digestive tract, and then the very slow transit near the end. You have high acid in the stomach, and this decreases or increases to neutral pH when you get to the large intestine. It is the small intestine and large intestine are anaerobic and have a large surface area. If we look at a microscopic blow-up of gut structure, it looks like this. You have this thin layer of epithelial cells. They are covered in mucus. And there's a presence of a large number of immune cells and tissues. There are dedicated regions of lymphoid tissue in this area, and these are called pyropatches. And of course you'd have these because this is probably one of the largest concentrations of microorganisms that your body's going to see. The role of this single layer of epithelial cells is to uptake nutrients, to secrete mucus to protect the layer. It produces antimicrobials. It also produces some enteric hormones. And then also there's a rapid cell turnover to get rid of any organisms that you don't want that are attaching to the intestinal mucosa. There are multiple gut functions in health. There is a nutritional function, right? There is the immune function, prevention of allergies and prevention of infections. And then there's neurological function. That is regulating the amount of energy that we're using based on how much nutrition we have. It turns out there might even be some mood regulation. The gut microbiota affect gut function in all of these different areas. So what are the roles of the gut microbiota? First of all, it's barrier protective. They prevent pathogens from binding by pathogen displacement. They're there, so pathogens can't bind. They compete for nutrients with the pathogens, and they produce antimicrobials of their own. There's a metabolic function. Cells in the intestines will actually take some of the mucin and other stuff, they will degrade it into butyrate. That butyrate then finds its way back into the epithelial cells, and that actually feeds those epithelial cells and powers them. They help degrade plant polysaccharides and mucus. They synthesize vitamins. Vitamin K, biotin, folate, and B12 are all taken up by humans from their gut microbiota. And then finally, there's the immune function. They educate the immune system, and they will induce the production of antimicrobials by the host. Let's look at the metabolic role of the GI microbiota. Again, here is another version of that community physiology that we've talked about. You have complex polysaccharides. They will get broken down. Some of them will get broken down by various species. And these simple sugars, lots of it is absorbed by the host. Some of them are then still used by the gut microbiota and changed into short chain fatty acids. Some of these will get absorbed by the host. Now, this is different than what we talked about with ruminants. In ruminants, this is their main source of nutrition. With us, it's a side amount of nutrition, these short chain fatty acids, acetate, propionate, butyrate. We will absorb them, but it's not our main source of nutrition. There is also secondary fermentations by methanogens, sulfate reducers, acetogens, propionibacteria that will produce propionate, acetate, lactate, etc. that again can be absorbed by the host. What I want you to get from this slide is to understand that a community in the gut is degrading the polymers that are there just like they do in every other environment, and then some of it is absorbed by the host. And secondly, that this is a benefit to the host, but it's not the major source of nutrition for humans. Okay, time for a quiz. If you look carefully at the data, you will notice that each individual has their own unique flora. What I want you to think about is, how do you explain this? Everyone has a unique flora. How do you explain that? What do you think is the most important thing? Now, you'll notice I haven't made this a quiz for SCORM that's counted, because it turns out there's a couple different answers. Many people would say A, and there is evidence for this, but C also plays a role. And of course, the microbe fairy is always a good answer. So what really is it? So there was an interesting paper that came out where they looked at bacterial phyla in a group of people, and they're all listed here. Each one of these lines is an individual. And if you look at the phyla, they vary significantly between them. Instead of having a consistent phyla, it appears that a core gut microbiome exists at the level of metabolic functions. This conservation suggests a high degree of redundancy in the gut microbiome and supports an ecological view of each individual as a, quote, island, unquote, inhabited by unique collections of microbial phylotypes. Such as in an island, if you look at specific islands and you go to one specific island and you go to one that's maybe a few miles away from it, and you look at the organisms that are filling each niche, they'll be different, but every niche has an organism. And what this figure is showing you is the same thing. Well, the bacterial phyla are different. If you take the genes of each of these organisms and you compare them, and you look at what are called COG categories, which is just various functions, amino acid biosynthesis, degradation of polysaccharides, etc., you find that it's very consistent across that. So each organism in the bacteriophylla is filling a niche. And once that niche is taken, other ones often can't outcompete it, and that's why you get the uniqueness. This leads to a very interesting dietary subject. There is a very frustrating aspect to weight loss. Any dieter will tell you that while it is possible to lose weight, it is just as much of a challenge to keep it off. Our bodies seem to want to stay at a specific weight, a set point, and anything we do that may deviate from that weight, our physiology fights. There is one insidious exception. If we overeat for a long enough period, our bodies will raise that set point to a new higher number. When a person takes in fewer calories than they burn, they initially lose weight, but after a while this progress slows. The body misinterprets this weight loss as starvation and begins to conserve energy and burn fewer calories. For most of human history, this mechanism was essential for survival because food was often scarce. But in the modern world, it can be a curse. Once an individual reaches their weight loss goal and goes off the diet, this metabolic conservation continues and they burn fewer calories, making it much easier to regain the weight. A recent article in the New York Times provides a stark example of this phenomenon. They followed up with 14 contestants from the Biggest Loser TV show to see how they fared 6 years later. As you can see, the results are discouraging. 13 of 14 contestants had increased in weight, with 3 of them weighing more than they did before the contest began. Even worse, their metabolism never recovered, even 6 years later. All the contestants burned fewer calories each day than before the weight loss. One contestant, Erin Egbert, was successful. Why? She maintained her efforts at weight loss and accepted the handicap of having to deal with a slower metabolism. Erin made a lifestyle change. She didn't start and stop a diet. So this is all fascinating, but what does it have to do with the human microbiome? The human microbiome may have a role in this metabolic change. In 2016, Face et al. Wanted to investigate this weight gain after weight loss phenomenon, but first they needed to find a model system and settled on mice. Can they see the same phenomenon? Let's set the stage. They had 2 diets, a normal chow diet, NC, that keeps a mouse at a healthy weight, and a high-fat diet, HFD, that makes a mouse gain weight and represents a poor typical diet in the US. The mice were split into 4 groups. NC, that consumed normal chow the entire experiment. HFD, that consumed the HFD, the high-fat diet, the whole time. Prime HFD, that started with the NC diet and were exposed to the HFD diet from 7 to 12 weeks, and then finally Psych HFD, that went through 2 cycles of HFD consumption, 0 to 4 weeks and 7 to 12 weeks. The thought was that if you cycle through the diet, this represents the yo-yo dieting that a lot of individuals do that are trying to lose weight. What you can see from the figure is that the Psych HFD mice gain weight faster in the second round of the high-fat diet, even in comparison to the mice eating this diet for the first time, Prime HFD, shown in green. The graph on the far right shows that this difference is statistically significant. So yes, they can replicate the results seen in the Biggest Loser contest using mice and this experimental protocol. Further investigation showed that initial weight gain causes a shift in the microbiome. Pictured here is a primary coordinate analysis that statistically represents all the members of each mouse's microbiome and then plots them graphically. You don't really have to understand the underlying statistics. Just know that the closer the points are to each other on these plots, the more similar their microbiomes. Figure B
shows all the mouse microbiomes before the experiment began. And as you can see, they are all pretty similar and cluster together. After 4 weeks, the picture changes. The mice that consume the HFD, psych-HFD and HFD, have a profound shift in their microbiomes. After a subsequent 3 weeks of dieting, while the weight of the psych-HFD mouse has returned to normal, their microbiomes are now at an intermediate state and did not return to normal. Clearly, the microbiome is changing on the high-fat diet, but does this affect their more rapid weight gain? To answer this question, the experimenters did some fecal gut transplantation experiments. The microbiome of psych-HFD mice, those that had gained weight on an HFD diet and then lost it, was collected and transplanted into germ-free mice. As a control, the microbiome of an NC mouse who had never gained weight and ate normal chow the entire time was also transplanted into germ-free mice. Germ-free mice receiving the psych-HFD microbiome and fed normal chow did not gain weight. However, if the germ-free mouse with the psych-HFD microbiome was on a high-fat diet, the same rapid weight gain was seen as if the mouse had gone through a weight gain, weight loss cycle. The control NC mouse did not see this enhanced weight gain on either diet. It appears the microbiome is a significant cause of the more rapid weight gain. These effects were not observed in germ-free mice given a microbiome of a normal chow mouse, and further investigation showed that the metabolic activity of the microbiome of the psych-HFD mice is altering the metabolism of the mouse. Specifically, this was found to involve dietary flavonoids. Dietary flavonoids are found in high amounts in plants, and high-fat diets are often lacking in these compounds. Counterintuitively, a lack of dietary flavonoids causes a shift towards flavonoid-degrading bacteria. More than likely, these microbes do not depend on flavonoids as a nutrient, but instead are growing on the other nutrients in the high-fat diet, and they also happen to have the capability to degrade flavonoids. During a shift to a normal chow diet, the dietary flavonoids return to normal, but the altered microbiome degrades them. It turns out flavonoids in the diets influence the host's behavior. Other research shows that the presence of flavonoids decreases food intake and increases activity and energy expenditure. In the experiment here, the cycled mice had their flavonoids removed by their microbiome, thus moving less and expending less energy. Can you cure this by feeding the mice with an altered microbiome excess flavonoids? That was the next question. The research performed a test on the effect of flavonoids by including a fifth mouse group. This group went through the psych-HFD protocol, shown in red, but on the second cycle of the HFD, they were given excess flavonoids. When this was done, there was no excess weight gain as seen in the HFD group. Compare the red line in B to the mustard yellow and green lines. And it shows you that the mustard yellow gained weight just as the mice that had first been exposed to the high-fat diet. A flavonoid deficit appears to be the whole story. As you can see in the graphs E and F, the energy expenditure, which flavonoids are known to influence, is similar to the flavonoid-treated group as it is to the controls, and is much higher than the cycled group that was not given excess flavonoids. Could the same story be true in humans? Are flavonoids the answer? The Biggest Loser graph looks a little like the same experiment we saw in the mice. If the research applies to humans, it would mean that weight gain changes the microbiome and weight loss does not change it back. These folks are low in flavonoids, and this causes an increase in food intake and a decrease in activity. Is there any evidence to support this? Do obese individuals have a different microbiome? One of the early findings that came out of looking at gut microbiomes of humans was the claim that obese individuals have a different microbiome than non-obese individuals. A major idea was the ratio of bacteroidetes to firmicutes was high in lean people and low in those that were overweight. It was thought that firmicutes are better at degrading complex plant polysaccharides, thus making them available to the host. The microbiome of obese individuals had a microbiome that was allowing them to get more calories from the food that they ate. Several recent analyses call this whole idea into question. Z and Schloss recently examined 10 different studies that compared non-obese to obese individuals and looked at two common metrics that address the similarity of the microbiome between groups. Shannon Diversity Index is a measure of how many species make up your microbiome. The larger the number, the more diverse your microbiome. In previous studies, it was claimed that non-obese individuals had a more diverse microbiome, and a more diverse microbiome has been shown to be healthier. However, the data shown here indicate there is no significant difference between obese and non-obese individuals. The researchers also looked at the ratio of bacteroidetes to firmicutes, and again, found no difference between groups. This refutation of a popular conclusion demonstrates we still have much to learn about the human microbiome and how to analyze it. What does this say about the mouse studies? Well, different types of analyses were done, and more work needs to be done to ferret out if there are detectable differences between obese and non-obese individuals. I now want to switch gears and talk about a microbe discovered to have important effects on humans, Akkermansia municiphala. Research into the human microbiome is in its infancy, but scientists are already making a few intriguing observations. Almost all humans have Akkermansia municiphala as a resident of their intestine, as do many other animals. A municiphala is about 1% of the total population of the microbiome in humans, and specializes in mucin degradation. This relationship appears to be of mutual benefit. Let me explain how. MUC2, the mucin protein, is a dominant protein in the gut mucus. A municiphala degrades the mucin protein our body secretes. This protein has sugar added to it as it goes through the export process, and it ends up forming a lattice network as part of secreted mucus. It has a relatively firm structure, which is altered by A municiphala by proteolytic cleavage. This cleavage does two things. First, it frees up the mucin to be taken up by A municiphala as a source of nutrients. Thus, A municiphala is not dependent upon our food intake for survival. Second, it liberates the oligosaccharides in mucin, making them available to the microbiome community. Finally, the advantage for the mammal is twofold. First, the expansion of the mucus by degradation makes it a more substantial protective barrier. Second, some of the mucus is recycled as acetate, butyrate, and propionate that is taken up by epithelial cells and used to power their metabolism. We have also found Akkermansia municiphala correlates with good gut health. Patients who have known gut perturbations, such as those observed in Crohn's disease and irritable bowel disease, have a much lower percentage of A municiphala in their gut. Studies with mice having a known microbiome indicate that A municiphala impacts the functioning of the immune system and metabolic and signaling pathways. The addition of A municiphala has also been shown to reverse unhealthy effects of a high-fat diet. Mice fed a high-fat diet often become obese and develop type 2 diabetes. If Akkermansia municiphala is present as a probiotic in their chow, it mitigates these conditions. From what we know so far, Akkermansia municiphala seems pretty beneficial to humans. A municiphala is an early example of what we might learn about our own microbiomes. It may be possible someday to develop gut microbiome transplants that can reverse obesity, type 2 diabetes, or maybe even heart disease. The future is sure to be exciting in this field.
An emerging idea is that disease states are correlated with altered microbiota. The question is the altered microbiota a consequence of disease or a cause? Many common modern diseases may in part be due to changes in the microbiota. That is, parts of our microbiota, our old friends, change in some way and start to contribute to disease states. Some examples are the rapid increase in allergic, autoimmune, and inflammatory diseases, and the rapid increase in metabolic diseases. Some examples of gut disorders include inflammatory bowel disease, celiac disease, colon cancer, irritable bowel syndrome, and susceptibility to gut pathogens. It also appears that some autoimmune diseases and allergies involve the microbiota. Finally, the microbiota may contribute to diabetes, obesity, and mental disorders. I posted another video on the gut-brain axis, which I encourage you to watch, which is really interesting. In the last part of this lecture, we will focus on cardiovascular disease, an illness that killed 647,457 people in 2019 in the United States. What you can see is there's all sorts of metabolic and immune dysbiosis, and this can lead to excess calorie consumption, altered lipid metabolism, obesity. It causes an increase in risk for markers for cardiovascular disease. You have hypertension, etc. So there's all sorts of things that can happen from contributions to gut microbiota. One thing that we're going to focus on, which really has clear indications, is TMAO, which is trimethylamine oxide. And that has involved, it seems, in cardiovascular disease. And I will use the abbreviations TMAO and CVD. An important marker in heart disease is the production of trimethylamine and oxide. Those who have high levels of TMAO have a higher risk of heart disease. It turns out the gut microbiome contributes. Phosphatidylcholine is present in our diet in cheese, shellfish, eggs, and meat. Our body degrades this to the fatty acids in choline. Members of the gut flora are then able to convert the choline to TMA, and this is then oxidized by our livers to TMAO. The presence of TMAO has been shown to increase arteriosclerosis. This leads to heart attacks, strokes, and death. Recent results also show that carnitine is metabolized by the gut microbiota to TMA, further promoting arteriosclerosis. This TMAO accumulation is not seen in vegans fed a steak dinner, clearly showing that differences in microbiota will have a different result. It also indicates that vegans must have a very different microbiota than meat eaters. Also, if you suppress the gut microbiota in meat consumption and you feed them a second steak dinner, you again see no effect, and this is shown in the figure on the right. Here is the initial amount of TMAO after being fed a steak dinner, right, of these individuals. You then suppress their microbiota by giving them antibiotics, feed them another steak dinner, the TMAO goes away, and then finally you let the microbiota recover, you feed them another steak dinner, and it comes back. Thus, modulating the microbiota may have an effect in decreasing the amount of TMAO and hopefully heart disease, and that's an area of interest in research. As a last thought, I wanted to say a few things about probiotics. Probiotics are pure cultures of microbial strains that yield a measurable health benefit when tested in controlled trials. There are also prebiotics, and these are foods you eat that feed your probiotic microbes. At first, there was skepticism about the effectiveness of probiotics, but as they have been investigated, scientists have realized they can have beneficial effects. This is a very young field, and lots of stuff you see on the internet where snake oil salesmen hawk their probiotic miracle cures is obviously baloney. However, probiotics have been able to show positive effects in some illnesses. Some probiotics enhance function of epithelial barriers, and others appear to modulate the host immune response. Okay, that's it for host microbe interactions. See you next time.